All right. In the last video, we looked at prophase one. The whole entire prophase one is very eventful. Eventful is in that it works towards genetic variation. There must be genetic variation. And how does it accomplish that? It accomplishes it via the process of crossing over, right? Crossing over, basically your homologous chromosomes will be next to will lie next to each other, forming a bivalent. Now the inner chromatids of your homologous pairs will overlap one over each other, and then they'll perform a process called crossing over. As they overlap, that overlapping is called chiasma, right? Many chiasmata, right? Now this brings about your genetic variation in cells. Then after that, the cell basically will go into metaphase I. What happens during metaphase I? During metaphase I, homologous chromosomes basically will move to the equatorial region of the cell, to the equator, right? Then they will lie parallel to each other. The homologous pairs will lie parallel to each other, each on the opposite end of the equator. You must mention that there is an equator, right? Then what will happen? Now they will lie in such a way that it is random, like your homologous pairs will lie random. It does, as to which one lies on which side of the equator, it is completely random. That is a random arrangement of chromosomes, which brings further genetic variation. So the first genetic variation that we see is brought about the process of crossing over. And then the second one is during metaphase, during random arrangements. They're basically going to lie randomly on the opposite sides of the equator. Then the spindle fibers now prepare for what? They will prepare for anaphase, because they are attached to the centromere. They will begin to shorten in anaphase. As they shorten, they pull homologous pairs to the homologous pairs apart. Each chromosome, each whole chromosomes, might I add, will be pulled to the opposite pole of the cell. Then this separates homologous chromosomes to each pole, and then the cell enters telophase. Obviously, where new nuclear membrane is formed, a nucleolus will now start to reappear over here, and then cytokinesis takes place. Then each cell at the end of telophase one will have half the number of chromosomes, and then after having half the number of chromosomes, it will have genetic variance due to those two processes: random arrangement during metaphase one and crossing over during prophase one. Right. Now I want us to go back here, right? I want us to draw them. Now let's use our own. Good. Remember last time we ended here by prophase one, and then crossing over took place, chiasma, and then we essentially during your late prophase, you will have a cell which has your nuclear membrane disappears. Let's just let it go. You'll have a cell that has the following chromosomes. Shorter ones, these are the homologous pairs with a bit of genetic variation there. And then these ones, the taller ones. There. So that's basically what you're gonna have. And then this prepares us for what? It will prepare us for metaphase. Now, what happens during metaphase? That's what we need to look at. Let me see here. My pen is not working. Right. During metaphase, Remember the spindle fibers, right, from the centrosome or centrioles will now attach to the centromere of each chromosome, right? As these chromosomes now start to move to the equatorial region of the cell, right? And then by metaphase, you'll find that all of the chromosomes are now at the equatorial region of the cell. So let's move to metaphase. Metaphase I. First things first, during metaphase, we have an equator or a metaphase plate. That's the equatorial region of the cell. Now, what will we have on the equatorial region of the cell? We'll have 1, 2. Remember, these are replicated chromosomes. Let's not forget. And you have a bit of that. You have a bit of that. And then on the opposite end, and we have this. Now, this is your metaphase. During metaphase, what is important for you? You must mention that there is an equator, right? On this equator is where your homologous pairs will lie randomly. So, randomly. So this is random arrangement of homologous pairs during metaphase I that brings about genetic variance. Good. And then each of the chromosomes will lie on the opposite side of the equator, each of the homologous pairs. Not each of the homologous pairs, but each chromosomes from the homologous pair will lie on the opposite side of the equator. What are we missing here? We are missing the spindle fibers that will attach to the centromere of each chromosome. So that's basically what happens. And then in Anaphase I, the cell is ready for Anaphase I. In Anaphase I, a whole chromosome will be pulled to opposite ends of the cell. So these chromosomes are now being pulled. It's a tug of war. Take note of which chromosomes are pulled to which poles. We often make that mistake of not knowing which one gets to be pulled to which side. Let's see. We have this one, first one, that's going to be pulled to that side. And then the green one will be pulled to that side. So let's draw the green one over here, slightly longer. Centromere. Good. And it has some genes from the other one. And then obviously this side, you'll have this chromosome being pulled to the opposite end of the cell. And then the blue one will move together with the yellow chromosome. Let's use colors. I think it makes sense. There's a bit of that. Good. Now, they are being pulled. Remember, the arrangement was basically random. Now, this pulling of chromosomes. So what are you going to say? Chromosomes are pulled to opposite poles of the cell. Now, what is happening here? Homologous chromosomes are split. Chromosomes are split apart. Remember, they were homologous chromosomes. Now they are being split apart to different poles of the cell. When the chromosomes separate, this separating of chromosomes, this is known as your karyokinesis, the splitting apart of your chromosomes to opposite poles of the cell. Remember, in the next phase, you'll see cytokinesis, the splitting of the cytoplasm. Here's the splitting of the chromosomes. So chromosomes are being pulled to opposite poles of the cell. Okay? So we are splitting what? We are splitting your homologous pairs. Now, how many chromosomes do we have now? We have 4 chromosomes in anaphase. It's important to always keep a record of the number of chromosomes that you have. It will make sense right now. During telophase. Now, during telophase I, here's what happens. Yay. During telophase I, if it's in an animal cell, you'll see something like this. My kids love this. They laugh every time I mention it. The cleavage furrow. Yeah. And then what happens? The nuclear membrane starts to appear again. And then the nucleolus. So what do you see? You're going to see the nucleolus starting to reappear. Nucleolus and the nuclear membrane. They start to reappear. Good. And then what will you see on opposite poles of the cell? You're going to see the following. On this cell, start with this one. You're going to see the red chromosome because it was pulled to this pole. Right? The red chromosomes with a bit of the blue chromosome in it. And then we're going to see this chromosome again. The green chromosome. Remember, it's a whole chromosome on this side. And then on the other side, the yellow chromosome was pulled to the opposite end with a bit of the green chromosome on it. And then lastly, there was your blue chromosome, slightly smaller, with a bit of the yellow chromosome in it. So basically, this is what you end up with. Good. And now those chromosomes, that cell splits completely during cytokinesis. Cytokinesis happens and the cells split completely. Now you have 2 cells. Now you have 2 cells. That is the first thing you notice after meiosis 1. We are accounting for things like 2 cells. There's a reason why I leave space there. You'll see just now. Now, let's account for these cells. Now you have 2 cells. Let's draw the chromosomes again quickly. Then the green one. There and there. Then this side you'll have the small blue chromosome with a bit of the red chromosome, and then you'll have a tall yellow one with a bit of the green one. There. This is basically what we drew. I made a mistake here. You don't have this, you have this. Perfect. Now we corrected that. Good. Now, notice how many chromosomes do we have in each cell? We have 1, 2 chromosomes, 1, 2 chromosomes. So the amount of chromosome has been halved. This means we have 2 haploid, represented by N. 2 haploid cells. 2 haploid cells have been formed. So there were, meaning there was halving of the number of chromosomes during meiosis I. There was halving of the number of chromosomes. Now these cells, these two haploid cells, they have genetic variation, meaning they're genetically different. If you look at the chromosomes found in each cell, it's completely different. So that's what happens during meiosis I. You end up with two haploid cells because there was a halving of chromosomes during the cytokinesis in Anaphase I. Then, due to a random arrangement of chromosomes and particularly crossing over, now these two cells are genetically different. Now, great. So that is your meiosis I.